My great pleasure to introduce our research, especially focused on the bioresponsible smart drug delivery. So the story is like, let me tell you something like this. 11 years ago, once I went to the U.S. Embassy office in Shanghai, in China, to try to get the visa, and the immigration office asked me, what's your future research focus? And I said, drug delivery. And he double-checked me. It's like, oh. Okay, and later I needed to explain what's the meaning of the drug delivery. Okay, so, and later I told him it's like the drug delivery is like really, deliver this kind of therapeutics to the body, to the specific sites at the right time with a relatively correct dose to help enhance the treatment efficacy to help the patients. Okay, so this is really Breaking Bad. Okay, yeah, so you like this kind of TV show, right? Okay. So our lab is trying to integrate the biomaterials design, biomolecular engineering, and the micro-nano fabrication together to form this kind of versatile formulations. And we call it I medication. I means innovation, means inspiring, means the patient-centered medications. Okay, so potentially we really want to develop this kind of new systems for treatment of the diabetes, cancer, and for the regenerative medicine applications. So especially we are focused on the functions of the different drugs, just like this kind of a magic cube. The smart is our, the keyword of our research. And the magic cube, we have 6 phases. Okay, so the purpose of the magic cube is to normalize each phase. Okay, however, it's really challenging for me to do that. Okay, so I just want to randomly play with it to form the different patterns. And here means to apply this kind of different drug delivery systems for the different applications to form the different patterns. Okay, so especially focus on functions of the different therapeutics, and we can add some specific materials and formulations to encapsulate them, and to make them smart, we can add some stimuli triggers. Okay, and they can be some physiological signal-based triggers like a pH level, like enzymatic activity, redox gradient, glucose ATP levels, or external-based triggers like a photomagnetic, electrical, even like a thermal or mechanical force. They can also provide the triggers to make sure the drug can be released at the right time, right places with a relatively correct dose. So I will show you more examples later. Okay, detailed examples later. So as you probably know that in our body, always associated with some specific diseases or specific organs, tissues, the physiological signals or biomarkers, the concentration usually is different from the normal tissues, from the normal organs. So then we can leverage or we can take the advantage of such concentration difference or gradient of the biomarkers to promote the drug delivery. So this is today's focus. For example, in the tumor site, especially the pH level is a bit acidic. So then we can develop some acidic sensitive material which can be degraded or dissociated in the acidic environment and form some nanoparticles, load the drug inside, and later on deliver into the tumor sites, and they can be dissociated or dissolved in the acidic environment in the tumor site and to promote the drug release. And in the wound sites, typically also it's a bit acidic and also some enzyme can be highly expressed. So we can also leverage this kind of environment to trigger the release of the drug. And blood glucose level certainly is a very important biomarker, to trigger the release of the insulin for treating type 1 and advanced type 2 diabetes. So I will show you more details later. This is not quite sensitive actually. Right? And today I will focus on 2 topics. One is to form the synthetic pancreatic cells or engineer the smart insulin delivery systems for type 1 and advanced type 2 diabetes treatment. And the other side is so-called programmed nanomedicine to promote the drug release or sequentially deliver 2 or multiple drugs, especially to the tumor microenvironment or to the subcellular environment to enhance the treatment efficacy. So I will talk about the diabetes treatment first. So as you probably know that diabetes is a global burden currently affecting around 400 million people, and every 6 seconds one dies from diabetes. And this number will be further increased to over 600 million. And at that moment, 1 in 3 Americans will have diabetes. So it's a really challenging issue. And just some background about the insulin and diabetes. So insulin, this is a hormone produced in the beta cells in the pancreas. And the active format of the insulin actually can bind to the insulin receptor on the cells, especially from the muscle, liver, and fat tissues. And just like a key to open the door, let glucose go inside to cells to perform the downstream metabolism, okay? And then, glucose can uptake by the cells from the serum, okay, inside the cells. And however, for the type 1 diabetes, the patients actually can no longer produce insulin as they need due to the autoimmune reaction. Actually, the beta cells which produce insulin damaged, okay? They never work. Then, for the type 1 diabetes patients, they need additional insulin. And also for type 2 diabetes patients, they can generate insulin. However, it's not sufficient. So they also need the additional insulin, especially for the advanced type 2 diabetes patients. And as a traditional treatment method, we call it the open loop-based method. The patients need to frequently monitor their blood sugar levels using this kind of electrochemistry strips. Okay, people call it like very the pesky strips. Okay, from type 1 diabetes patients, and then perform the multiple injections every day. This is like a 2-week supply of syringes. Okay, so it's really painful. Okay, and overdose is fatal, can cause hypoglycemia if you inject too much insulin. And also, it's really hard to tightly control the glucose levels, them to perform this kind of insulin injection. So as a result, many diabetes complications will occur. However, in the healthy body, the insulin secretion level, you can see the blue line, always associated with the tightly, follow the red line, okay, the glucose level. So red line glucose level, blue line is like the insulin secretion level. So they have this kind of very tight relationship. Because the beta cells are naturally smart, they can sense the blood sugar level and secrete the insulin at the right time with the correct dose. Okay, so very smart. Okay, and based on this, people naturally thought about that. How about developing the closed-loop insulin pumps or artificial pancreas? And typically involve 2, devices or 2, components. One is a sensor which implanted to the body, can sense the blood sugar level, and then pass the signal to the insulin pump. And after certain calculation, the relatively, the certain dose of insulin can be, infused to the body. Okay, so this is the general mechanism. However, FDA always has concern about this kind of system. Even though, like last year, the first artificial pancreas just approved by FDA, but still, like many issues there, and the FDA still have certain concern because it's really hard to achieve the accurate signal feedback. The patients still need to do this kind of calibration every day, especially before or after the meals, okay? And then the second thing, it's really hard to prevent biofolding. So after a couple of days, the patients still need to replace the sensor here and the needle here. Otherwise, the fibrosis will grow. Some tissue will grow around the sensor or also the needle and affect the performance of this kind of insulin pump. So still many issues here. And recently, people become more and more interested in developing the synthetic artificial pancreas, just using the synthetic materials or modified insulin to achieve this kind of smart insulin delivery. So the general motivation is like this. Just think about that. In our body, we don't have any electronic devices, any electronic chips or wires, but they can still control the glucose level very well, all based on this kind of biochemistry process, the chemical reaction. So why not, mimic this kind of process and to achieve this kind of function, just using the synthetic materials and involve some biochemistry process and to achieve the smart insulin delivery in a biomimetic way. So this is the motivation. And typically involve two key elements. One is the factor that can sense the glucose level, and the other one is material that can be activated to release the insulin at the right time. Especially once the blood sugar level goes high, more insulin can be released from the system. And once the blood sugar level becomes normal, less insulin released, or just like a basal release. Okay. So here is a summary table regarding the current development advances, from these kind of pharmaceutical companies as well as research labs. And you can see many pharmaceutical companies very interested in developing the smart insulin systems. Okay, we call it synthetic systems. Like Merck, they actually just announced that they have already started the phase 1 clinical trial like 1.5 years ago. And like Eli Lilly, Sanofi, Novo Nordisk, they have the specific research groups focused on this kind of smart insulin delivery systems. And also research labs like our lab from the joint BME departments between the UNC and NC State, and also like MIT, Bob Langer, Dan Anderson's group, okay, and like a Caliber Institute, okay, in the San Diego. And our lab is trying to develop this kind of new materials, new formulations, as well as new devices, and try to potentially address this kind of problem to make sure the patients can eat whatever they want to eat and whenever they want to eat. Okay. So next, I will briefly show you several examples recently developed by our group. Okay. So we are pretty interested in using the glucose oxidase, and this is kind of an enzyme, a glucose-specific enzyme. Enzyme is like a protein, catalyte, okay, to do this kind of catalysis reaction, okay. And this enzyme actually is like just in the glucose sensors, the monitoring systems, okay, because this enzyme can convert the glucose into the gluconic acid, okay, to promote this kind of reaction, to generate the acid, gluconic acid. So then, we can involve some acidic sensitive material or acidic, degradable material there. And once the blood sugar level goes high, the local acidic environment is generated. And then, if you involve some acidic sensitive material there, and they can be dissociated or degraded or swelling or shrinking, then pushes the insulin release outside. So this is the general mechanism, okay? Yeah, so for example, we apply the chitosan as a major material. As you probably know that chitosan is a polymer, it's a polysaccharide or polysugar, originated from the shells of the shrimps and the crabs, okay? So kind of biocompatible, okay? And so I'm still not sure this shrimp is the true shrimp or not, okay? It looks very huge, okay. And later apply the electrospray method to form the quite a uniform structure, the microgels, okay. And in the high glucose level based solution, such material actually can be charged, okay, because the hydrogen ion, the acidic environment generated, then the hydrogen ion will bind to this kind of material. Then the polymer chain will be charged. Then each polymer chain will be repelling to each other, right? Since it's like a positive charge, both chains like a positive charge, then they should be repelling to each other. Then the whole system will be swelling like this. And then the insulin can be promoted to release outside, okay? Compared to the normal glucose level, like 100 based or like PBS only, okay? So this is one example. And one of my friends later on commented on my Facebook, quite small, smart sponges swell in super sweet solutions. So many S here, and I like it, okay, yeah. So, and the next example is we call it nano network. And this one is like, it looks like just like a nano network, it's part of the nano, composites inside, okay. Yeah, it's cross-link structure. And if you look into detail, it's like each one is like a, nanoparticle, like this. But overall, it looks like toothpaste. Yeah, looks toothpaste. but they can be injectable underneath the skin, can stay there underneath the skin. And this material can be acidic degradable. In the acidic environment, they can be dissociated, and insulin can be promoted to release outside. And we also test in the type 1 diabetic mice, and only one injection of such nano network can maintain the blood sugar level. This is the blood sugar level in the normal range below 200 up to 10 days. For some of the mice can even maintain their blood sugar level in the normal range up to 2 weeks. So here I just want to clarify. And for the human beings, usually the normal glucose level is below 100, and the mice below 200. So this is, the difference, okay? Below 200 for mice is normal, okay? And if you get rid of the enzyme, just put the insulin in this kind of nano network and inject it to the body. And on the second day, the blood sugar level has already been quite high. You can see this blue line, and then gradually increase. So you can see the significant difference here. And this one actually was featured on the ACS Nano, the American Chemical Society Nano Journal, as a cover. So I just show you 2 examples. However, we still had several challenges to be addressed, and they are actually kind of our ongoing focus, especially like how to achieve the faster response. Okay, the pharmacokinetics similar to the normal beta cell activity, how to avoid the hypoglycemia. If unexpected large amount of insulin released outside, how to avoid the hypoglycemia. So probably co-delivery glucagon. Okay, so we have already recently invented one formulation for this one, and will be published very soon. Okay, and sort of things like how to realize easy of treatment, further reduce the injection times. Okay, or use the microneedle array painless-based treatment method. So I will later on, emphasize on that. And the fourth thing is like how to solve the biocompatibility issue without any in vivo toxicity and no long-term side effect. Especially for the first one, how to achieve the fast response. The PK is similar to the normal beta cell activity. Okay, so I just show you 2 examples, and they can respond to the, the high blood sugar level. However, the response speed later on we found that it's not that fast. Yeah. Then you may ask, how fast about the beta cell activity? I will show you one picture. So I like this grape very much. Hopefully everyone like it. So as long as you like it, okay, you saw this picture, without eating the grape, your insulin level has already been increased. So that means this is a neuron associated, not only directly respond to the blood sugar levels, but also respond to the feeding, okay? Yeah, so it's really hard to achieve that fast response, like a neuron associated, okay, using the synthetic materials. But we really expect that using the synthetic materials, using the modified insulin. So once the blood sugar level goes high, after 10 minutes, 15 minutes, then insulin can be quickly released outside, then help reduce blood sugar level. That's enough. We don't expect that neuron associated that fast, but 10, 15 minutes enough. But we don't expect that like after 1 hour or 2 hours, then insulin released outside. Then it's too late, okay. And in the past, okay, this kind of idea using the enzyme to achieve the glucose responsive insulin delivery, this kind of concept actually, conceived in the 1980s. Okay, that means 30 years ago. And usually people apply this kind of reaction to achieve smart insulin delivery focused on the right side of this reaction. So just as the 2 examples I show you, okay. And they developed this kind of different materials, different like formulations, try to, show this kind of fast response to the acidic environment. However, later on we found that our body, the life system, is a buffer system, B-U-F-F-E-R, with many salts inside. So it's really hard to reduce the pH level, reduce the acidic environment to gentle acidic environment within a shorter period of time. Like a pH level, neutral pH level is 7.4, 7.2. And it's really hard to reduce to 6 or 5 quickly to trigger this kind of response, okay? Yeah, so this could be a big problem. Although we can show this kind of response in the bottles, in the tubes, But once to the mice, to the animals, we found that it won't work that good. Okay, then we thought it could be some like issue, like as I said, our system is a buffer system. It's really hard to quickly respond to the acidic environment or quickly to reduce the pH level. So then we thought about how about change mechanism using a different way to achieve the potential faster response. And one day we noticed on the left side, actually the oxygen level reduced. Okay, not only generated acidic environment, and this guy actually consumed. And then we thought about that, how about using the hypoxia sensitive material instead of using acidic sensitive material to potentially achieve the faster response. So, by the way, so hypoxia, that means with a relatively reduced the oxygen level. Okay, this is so-called hypoxia. Okay, so we just want to change the mechanism a bit. Okay, focus on hypoxia environment material to trigger the release of insulin. So then we developed this kind of system. So it's a bit complicated. So I just want to make things simple. So basically we apply the polymer hyaluronic acid as a major material, and hyaluronic acid is also a polysugar, very biocompatible in many like plastic surgery as an implantable material inside. Okay, so they can be biodegraded, or also it's like show very excellent biocompatibility. So we apply this kind of material as a major material, and we modify this material with some hypoxia-sensitive components. Okay, then this kind of structure actually can form this kind of vesicle or nanoparticles inside the aqueous solution in the water, and then we can encapsulate the insulin and the enzyme inside. And then once the blood sugar level goes high, on the hypoxia environment, this kind of structure can be dissociated or dissolved in the water, and the insulin can be promoted to release outside. And at that moment, we don't know if this kind of system is faster or not. But later on, we demonstrated that it's extremely faster compared to the pH responsive system. Okay, and to make the treatment method easier, we put them into this kind of microneedle array. And you may ask, why microneedle array? Certainly they are very small, very tiny, okay? And the height here is about 600 micrometer to 800 micrometer. And the neuron cells underneath our skin is typically 800 or 1 millimeter away from the surface of skin. So that means you apply this kind of microneedle array to the skin, and they can't touch the neuron cells that significantly. So that's why it's painless. But you can still feel something, just like a mosquito bite you. Okay, that's it. Okay, compared to the general metal-based needles, it's painless. Okay, much painless. And this picture actually later on also highlighted by Science, like in the end of the 2015 as the top 10, research images of the year. And this kind of formulation originally is like quite compact. This is the electronic microscope image. Okay, of this kind of vesicles. And in the high glucose level-based solution, they become dissociated, degraded, and eventually disappeared. Okay, and later on, we apply this kind of mold-based method to form this kind of uniform structure of the microneedle. And the major material here is also hyaluronic acid, as I just mentioned, very biocompatible. And once I talk about the microneedle, I always like, showing this picture. And you can see the selling point of this picture is not this guy, actually this guy, okay? Because he's the second one, okay? Waiting there, quite nervous, okay? Other people still like laughing, smiling because they are not the second one, okay? So potentially, using the microneedle array, you don't have to worry about you will be the second one waiting the line like this, quite nervous. So psychologically, much better. Also, we tested on the mice, and such patch can easily penetrate the inside to penetrate the skin, and they can quickly reduce the blood sugar level from a high level, like over 550, to like a normal level, around 200, and it can maintain there for a while. And such reduction happens like within 30 minutes. Okay, so it's really fast. Yeah, and if you put the pure insulin without the glucose sensing system just the insulin in the patch, and you can see the blood sugar level, the orange line, quickly go down and quickly go up. So you can see the significant difference. And you can also adjust the enzyme and the insulin level in the patch, okay? For example, the red line is like with a half enzyme. So that means the reaction rate is not that fast, okay? Or not that, dramatic. Then, the response behavior is also not that significant, okay? So that means we can potentially, based on the patient's glucose level or diabetic condition, to develop some specific patches for the specific patients and potentially achieve the personalized medicine. And we also tested this kind of study. We put one patch there, blood sugar level quickly go down from high level to a normal level. And then at this time point, at the point 2, we put another patch, the second patch there. And you can see the blood sugar level can maintain there for a while, then gradually increase. Can maintain there for a while, then gradually increase. Yeah. However, if we, at this time point, if we put a pure insulin patch there, and the blood sugar level can further go down. Yeah. Because they just, pass through release the insulin, that's it. And the other one is like a glucose responsive one. They think the glucose level has already been okay, don't have to release. But the pure insulin one can further go down and to achieve the hypoglycemia. Okay, that means blood sugar level too low. Yeah, sometimes people may die. Yeah, here some might die. Okay, so it's quite dangerous. And so we create a smart insulin patch. Not only can quickly reduce blood sugar level within a shorter period of time, but also can potentially avoid the risk of hypoglycemia. And this is another story. And this time we combine the live cells, live beta cells, together with the microneedle. And here the microneedle can serve as a bridge to link the physiological signal in the body and to pass this kind of signal to the cells on the top of the microneedle. That means externally positioned cells. You don't have to put the cells in the body. So probably some of you know that for the type 1 diabetes treatment, besides the insulin injection, another way is like a cell therapy, okay? And this kind of concept actually conceived in the 1970s. Okay, people try to encapsulate the cells, the beta cells, like from donors, for example, or now currently it's like from stem cells, derived beta cells, in the microgels, capsules, and later on encapsulate to the body, implanted to the body, okay? And the nutrition can go inside and insulin can release outside. Glucose can go inside and insulin can go outside. But the things like the concern is like after like a couple of days, couple of weeks, and this kind of cell can die, because, some like fibrosis, some tissues will grow around this kind of capsules, okay? And you need to get them out and to put a new, capsules there, just due to the immune response. And now we just want to avoid this kind of issue. Just put the cell outside body and using the microneedle as a bridge to link this kind of signal together. So here is just a simple demonstration of kind of a proof of concept. And currently we are still like trying to further optimize this kind of system and potentially translate it. And this is our latest work, and will be published like next month. And the basic idea is using the red blood cells for the smart insulin delivery. And here, actually, we simply modify the glucose into the insulin, okay? So we just let the insulin carry the glucose molecule. And later on, we found that once with the glucose, such insulin can bind to the red blood cells with very high density, okay? Due to the interaction between the glucose and the glucose transporter. Because on the red blood cells on the surface, many glucose transporters are, okay? Then glucose want to go inside, but unfortunately it's like blocked by the insulin. So insulin can stuck on the surface of the glucose, the red blood cells. And once the blood sugar level goes high, the glucose can competitively pass through the glucose transporter, then just get the insulin out, okay? Release the insulin, okay? Based on the competition binding, and then insulin can be released. So this is, kind of a new mechanism to achieve the glucose response of insulin using a relatively easy approach. Okay. So recently, our study for the smart insulin delivery attract certain attentions, and more importantly, we received many emails from, let's say, a number of emails from the patients, and they provide certain priceless, encouragement, and we really appreciate it. For example, this one from a mom of the type 1 diabetes patient, and this one from a type 2 diabetes patient. Okay, and here's a voicemail I want to play. Yeah.
I just want to say, ironically, that's why I'm kind of saying I'm laughing here, is when I was a kid, I questioned about a patch and was told, oh, no, we don't know how much insulin would come out in the patch. That's the problem with that. Really hearing about that through, people quitting smoking through a patch. I thought, wait a minute, if they can get something going in their body through a patch, what about insulin? So I wanted to say thank you for hearing that, for hearing me say that. I really appreciate it. And all you're doing, because this would make my life so much easier, so much better, and put less harm on my body than I've been putting it through all these years. That would be fantastic. So thank you so much.
Yes, and we really appreciate such a touching voicemail, and so the idea is like the dose control of the insulin is extremely important. So this lady actually previously had this kind of invention or dream, right? And later she thought it's really hard to control dose, and later give up. And then once saw our this kind of research news, and she thought it's like a new hope. Okay, so for the patients, especially for type 1 diabetes patients or the ones type 2 diabetes patients, sometimes they don't care what kind of insulin they apply, what kind of device they apply. They really want to live a normal, like how do you say it, the life, okay, just like a normal people. So that's why the delivery approach is very, very important, to make sure they can live like a normal people. Okay, yeah. And currently we are working with Sanofi, together actually with JDIF, Juvenile Diabetes Research Foundation, and try to potentially translate this kind of smart insulin patch. And the original grant actually supported by the ADA, American Diabetes Association, and I also mentioned later. Okay, and meanwhile we also run our own startup company called Genomics. Genomics, right? We changed the G to Z. Okay, and my first name is like initial is Z. Okay, yeah, Genomics, and trying to develop the new tools for the smart insulin delivery. Okay, and currently we just like obtain some investment, okay, and some news will be released probably in March, okay. And I would like to briefly also introduce my personal, motivation, okay, and also my play history, okay. So this picture I show my grandmother, okay, and she had diabetes for many years, and she has already passed away. And so 30 years ago, actually, I really like play in this kind of syringes, okay, after this injection, and I injected the different liquids to different, potatoes, tomatoes, okay, apples, anyway. And once I was in the primary school, I invented this kind of toothbrush, okay, especially for the lazy people. And we put I put the toothpaste to a syringe, and later combine with the toothbrush, and you can just easily push, and here's the tubing, okay, and the toothbrush will come out, okay, yeah. So for lazy people, and 5 years ago, and together with my postdoc advisor Bob Langer at MIT, we developed this kind of toothpaste like injection gel, okay, and I showed you before, and 2 years ago we invented the smart insulin patch, okay, and I would like to say fun and love is the origin of the innovation, okay. And if I want to put the second sentence is money is the translator of the innovation, okay. Yeah. All right, so due to time limit, I will briefly introduce our research on the cancer side, okay. Yeah. So especially we call it a program of nanomedicine. So programming is always exciting, okay. This is firework, and after programming, you can form some patterns like this, okay, smiling pattern, and it could be more interesting. And this one is Kung Fu Panda, and after programming, this is just firework, And after programming, this guy may someday go to the moon, okay, yeah, gradually, okay. And for the drug delivery, the programming system could be also quite useful. Here's like actually quite complicated. I just briefly introduce. We can apply this kind of core shell nanocarrier, okay. It's like with a core and a shell, and in one nanocarrier, and we deliver two different drugs. One is a protein drug, another is a small molecule drug. Small molecule drug, we can deliver them to the nuclear because the most active site of the dogs, this small molecule drug, is in the nuclear. And the protein drug, we can deliver to the cell membrane specifically. So we can sequentially deliver them to the different sites of the cancer cells and enhance the treatment efficacy, not just simply mix them together and through to the cells. And then this kind of delivery efficacy could be quite low. And here it's like to perform this kind of sequential delivery. All right? So this is also cover, and we call it the UFO to deliver two different troops to the different site of the planet. Okay, and planet here is like a cancer cell. The file size is a bit large, and they can specifically, accumulate in the tumor sites and inhibit the tumor growth. Okay. And we also apply like a graphene to deliver two different drugs. Graphene is a two-dimensional, like a carbon-based material, and can also serve as this kind of delivery carriers. Okay. And we also design this kind of DNA nanoball. We call it a cocoon-like degradable DNA nanoball. Okay. So it's like we use the DNA as the building block material, and later on just self-assemble into this kind of cocoon-like structure. Okay. And we can load the anti-cancer drugs here, and inside the cells, they can be promoted to release outside, especially in the acidic environment. Okay, so we call it bio-inspired design. So just a cocoon, there's a worm inside with a lot of anti-cancer drug. Originally, this guy is sleeping, and inside the cells wake up and bite the cocoon and release the drug. Okay. Bio-inspired design. Yeah. And we also apply this kind of cocoon-like structure to deliver genome editing tool, deliver the Cas9 protein and the guiding RNA, and later on, this kind of genome editing tool can go into the cells, go to the nuclear, and to perform this kind of microsurgery to the DNA and change the face of the cells. Okay. Now we change it to meso. Okay. Yeah. To do this kind of targeted delivery. And we are also interested in using some new materials, potentially biocompatible, to perform the drug delivery. Like last year, we developed this kind of liquid metal-based nanomedicine. Using the liquid metal, the gallium indium, this is a law, and this one is not like the mercury. Okay, mercury is quite toxic. This one actually quite biocompatible. Yeah, and we just apply the ultrasound to provide the mechanical force and can quickly, smash them into the nanoparticles. So we can form the nanoparticles using a very easy way, and later we can load the drug to it. We can anchor the different ligand sites, and they can be easily delivered into cells. And into cells, they can fuse together. The outside layer will be dissolved and fuse together, and to promote the drug release. And eventually, they can be degraded inside the cells, especially in the acidic environment. So we create a nano-terminator, just like the movie Terminator. So originally it's like a bulk material, the bulk material, and later on they can form this kind of small nanoparticles inside cells. They can fuse together and eventually can disappear. Okay, terminate. Yeah. So by the way, we call it a nano-terminator. Sometimes U.S. writer, the news writer, reported like a nano-terminator, okay, and later on translated in Chinese, and then the title changed to cancer terminator, okay. So it's really awkward, okay, sometimes. And they said, okay, liquid metal can be the cancer terminator, okay. And then, we found that actually 4 stocks in China side associated with the liquid metal, all saturated, okay, after the news, okay, yeah. Anyways, this is just yeah. It's out of our expectation. We kind of changed the market, stock market, okay, in China. And later on, we found that Actually, such nanoparticles, once fused together, the X-ray contrast can be enhanced. So potentially it can be a diagnostic reagent, not only to treat cancer, but also can be an imaging reagent, okay? And the inside cells, they can fuse together to form the bigger one. And they can also tie it to the tumor sites, shrink tumor size. So as a brief summary of this kind of bioresponsible systems, and many factors we should consider for designing these kind of systems. For example, how to select the biological triggers, how to perform the response actions, and the material properties. And very importantly, we really want to eventually translate them, okay? So the translation criteria should be considered at the very beginning, like a clinical efficacy, biocompatibility of the material used, also the scaling up feasibility. And for the fundamental research, the design strategy is also important. Yeah. So next, I will briefly show you two actually design strategies we are interested. And especially the first one, bio-inspired. I have already shown you a couple examples, and we really like a bio-inspired design, okay, to get the idea from the nature and then create the naturally smarter systems. I will show you a few more examples, like we created the nanoplatelets. We extracted the membrane from the platelet, and later on coated such a membrane to the nanoparticles loaded anti-cancer drug, okay. So platelets actually serve as a very important role during the cancer development. Okay, actually platelets like a negative role. Actually can help the cells, transport to the other places. They can aggregate in the tumor site, and later on to help transport the cells outside and to develop this kind of metastasis. Okay, but now we can, make this kind of nanoplatelets and load the drug there. Okay, and they can still tie it to the tumor sites, but now they can release anti-cancer drug. And the immune system just think, hey, this is probably a fragments of the platelets, okay? And they can't easily find them, okay? Yeah. And they can also, catch the circulating tumor cells in the body to potentially prevent the metastasis, okay? Like this, they can shrink the tumor size and also prevent the metastasis. For example, this one is like to the lung. Original, the primary tumor is like a breast cancer tumor, okay. And this one actually our also latest study published in Nature Biomedical Engineering last Monday. And this time actually we directly apply the platelets as a carrier and to transport the immune checkpoint inhibitors, okay. It's like a PD-L1 antibody. So PD-L1 antibody or PD-1 antibody, we call it like a immune checkpoint inhibitors. Actually, very powerful cancer immune therapeutics. Okay, and previously actually cured Jimmy Carter, the former president's, the brain tumor. Actually, it's like a metastasis from the skin cancer. Okay, the melanoma, and this one actually such antibody can activate the T cell, and the T cell can attack this kind of cancer cell. But the only thing is like T cell activity can also, attack the normal cells, okay, clinically. If you just inject such antibody, systemically to the body, and the autoimmune disorder will happen, okay, clinically. So then we can apply the platelets to perform this kind of targeted or local delivery of the antibody. So how? And, after surgery to remove the tumor, after the removal, actually many patients may die even faster, okay? Even faster, why? Because the tumor can come back. If the residual tumor is there, micro tumor is there, and they can even grow more aggressively, okay? And during the surgery, some cancer cells come out and may develop this kind of metastasis, okay? And now, after surgery, we can inject this kind of platelets with the antibody. And the platelets actually themselves actually can target to the wound sites, to target to the surgical site. They can aggregate in the surgical site. And in the surgical site, in the wound site, they can be activated to form the small particles. The size is about 100 to 500 nanometer, and to promote the release of the antibody to deeper sites. Okay, and meanwhile, we also found that they can recruit the immune cells to enhance the cancer immunotherapy locally. Okay, so in the near future, we really actually want to translate this kind of technology because it's simple, and we can load the antibody to the platelets. Originally, the product can be from patients themselves or from donors, and later on separate the platelets and simply do the bioconjugation chemistry to load the antibody to the platelets, and later on re-inject back to the body, and then they can target it to the surgical sites and to perform this kind of immunotherapy, locally. Okay, and also to help recruit the T cell. And another thing is like others inspired. Some ideas can be from a totally different area. Okay, for example, we apply microneedle for diabetes treatment, deliver insulin, and now we can apply microneedle to deliver the checkpoint inhibitors, PD-1, PD-L1 antibody directly to the melanoma site. Okay, and to enhance the retention time of the antibody in the tumor site, and also avoid this kind of potential side effects, as I just mentioned, like autoimmune disorders. We can load the antibody to the tip of the patches, since the antibody is very expensive. Okay, so we can typically load it on a very small area. Okay, and they can significantly inhibit the tumor growth, this melanoma tumor on the mice, and enhance the survival time of the mice. And this is our recent study actually using the microneedle to deliver the heparin, a blood thinner, to potentially avoid this kind of clots or thrombosis in the body. Okay. All right, and as a chemist or researchers in this kind of area doing this kind of formulation in materials design, and sometimes the ideas just from the life. For example, from my wife's cooking, yeah. Because of food inspired. Okay, this one is like a quail egg encapsulated meatball, and the taste is different from the pure meatball or pure quail egg. Okay, it's quite delicious. Yeah, we have the insulin encapsulated microgels. Okay, we have the ants on the bush. We have the insulin neural network. We have this one is a bit complicated. Yes. We have the self-sampled peptide gel with the insulin, yes. And we have dessert, okay, layer by layer structure, and we do have layer by layer structure for control release. And we have already run 2 startup companies, okay. Run startup companies is not that easy. And the easiest way is to run a restaurant, probably. And potentially I may, run this kind of restaurant, combine the food and idea together. And if the students need some idea, maybe after dinner or lunch, may get some idea, okay? All right, I would like to thank my great students, wonderful collaborators, and terrific support from the funding sources, especially American Diabetes Association, the Pathway Award, the JDRF, Juvenile Diabetes Research Foundation, Sanofi, and seed grant from NIH, as well as the Sloan Research Foundation. Thank you very much, and I'm happy to take your questions. Yeah.